Okay, great. So thanks, Sarikadi, for the kind introduction. And also I must thank the organizers for giving me this opportunity to share this stage with all of you. And of course, I must thank Professor Biman Bagchi for conceiving the idea of this platform of SMCB and kind of encouraging all of us to sustain this activity. And this is really useful for our community in general. So it's good to have enthusiastic participation today. It's I find it rather exciting, so that so many students have participated, and following the norm of this seminar series, the significant part of my talk will be targeted primarily towards students. So we'll try to build the concepts. I'll try to establish how we think about the problems that we deal with. And then we will go along probably to more advanced results and so on. So as I promised in my title, we'll try to understand how do proteins talk to each other. Now that does not mean that today after the talk you'll learn how to talk to a protein, but at least I'll try to kind of give you the ideas that how to kind of address this kind of questions, and from a molecular thermodynamic point of view, and I'll try to elaborate upon that point. What do I mean by a molecular thermodynamic point of view? And of course, please feel free to stop me at any point if you have any doubts. You can also put in the chat box, and Sarikadi will help me with the questions. Now, so this is the question: how do proteins talk to each other? Now, when this protein communication becomes important, generally, whenever we, so when we are dealing with this kind of framework of biomolecular recognition and signaling. And such biomolecular recognition and signaling is a very crucial part of cellular communication and signaling processes. And to give you a more real life analogy, whenever you are using your sensory organ, whenever you are seeing something, smelling something, tasting something, there is some kind of an external stimuli, and that is kind of activating some protein or other on your cell. And when those proteins get activated, they need to pass on that information to some other protein and so on and so forth. For example, there is this very celebrated protein called GPCR. The structure of that got a Nobel Prize. And this GPCR is involved in a wide range of cellular activation processes, including vision. We'll talk about that vision-related processes in a minute. For example, there is a protein called rhodopsin, which is plays an important role in vision-related processes. And also, whenever you smell something, some small molecule will bind to this GPCR protein, and that protein will get activated. What do I mean by this activation? I'll come back to that point later. Now, once this protein gets activated, it needs to pass on that information, and then it has to activate some other downstream protein. So you can imagine it like an office floor or a factory floor where a lot of people are working, and some information or cargo will appear through the door, and then that information will be processed by somebody, and then that information will go on to some other desk, and so on and so forth. So even in our cell, there are these hundreds and thousands of proteins continuously talking to each other, processing information, and accordingly regulating all the activities, and so on and so forth. So as you can see, it's a very complicated framework of cellular communication and protein-protein communication. Of course, it may look very complicated and very difficult to understand, and it is. But being a chemist, we kind of resort to this cliched quote by Richard Feynman that everything that living things do can be understood in terms of the jiggling and wiggling of atoms. Of course, this is a bit of oversimplification. I mean, it's easier said than done, but we will try to actually take that approach and at least try to think how we can start to understand such protein-protein communication process. And as I said, these proteins, even though it may look very complicated, but these are nothing but molecules. They're essentially polymers made up of amino acids. And actually, they're very interesting molecules with very fascinating properties. And we'll try to explore some of those interesting properties, particularly in the context of this protein-protein communication, how they will pass on the information. So coming back to this question, how do proteins talk to each other? In an anthropomorphic view, if you think proteins were humans, one protein might say that, hey, I smell coffee, because the flavor, the molecule that corresponds to the smell of coffee, probably it's coming and binding to our olfactory center, and then that protein gets activated. And then it has to tell some other protein, and that protein also need to do its job and pass on that signal, as I said earlier. But of course, proteins cannot talk, really, right? So exactly what happens? So we might think of it like how, for example, two visually impaired or hearing impaired persons might talk to each other. So as you, of course, you know that two visually impaired persons might talk to each other by gestures, touch, and so on. So essentially, we are talking about some physical interactions or the shape, change in shape, and so on. So molecules do the same. They talk to each other by the change in the shape, molecular shape, the change in the interaction pattern, and so on. So by invoking this idea that activation essentially means the change in the molecular shape and the change in the molecular interaction pattern, and using that idea, we'll try to understand how proteins talk to each other. So now let's see that what is the molecular thermodynamic framework of doing that. And essentially, you have, all of you have seen this kind of diagram in your high school textbook, that you can always draw some kind of a free energy landscape or a potential energy landscape that you have seen. And mostly, these energy landscapes will have multiple minima. And traditionally, we can say, for example, call that one of them is a reactant. Let's say this minima corresponds to, let's say, the reactant R, and this, let's say, is the product P. And more often, I mean, normally what you need is you have to plot the free energy as a function of something called a reaction coordinate, right? So reaction coordinate is nothing but something that characterizes the change that is happening in the system. And what would be that reaction coordinate, how to characterize the change for a really complex process, that is one of the very challenging problems in our area of research, and I'm going to come back to that point. But just to highlight at this point, for example, we would like to study a polymer, long polymer chain collapsing, very similar to, let's say, the protein folding problem. Then radius of gyration of that polymer could be a very useful reaction coordinate, because radius of gyration is a measure of the effective shape of the molecule. So in the extended state, the radius of gyration will be large, and in the collapsed state, radius of gyration will be small. So that could be a very useful discriminator between the two states, let's say the extended state and the collapsed state. And the basic thermodynamics tells us that if we could somehow compute the free energy as a function of that reaction coordinate, in our case, let's say the radius of gyration, then if we know the free energy difference between all these the minima, for example, this minima corresponding to the collapsed state and the extended state, if I know this delta G, this difference between these two minima, then basic thermodynamics tells us what will be the population ratio of these two states, right? So essentially, if we know the free energy difference between different states or substates in some sense, then we'll be able to compute the population, relative population ratio between all these substates. Okay, so not only just now equilibrium population, the free energy surface can also give us very useful insight in terms of the kinetics or the rate constant. Although this is in a somewhat approximate sense, but at least basic transition state theory gives us this expression of the rate constant, where if you know the free energy barrier, this delta G dagger, which is essentially this barrier, let's say, if you can compute the free energy barrier, from there you get some rudimentary estimate of the rate constant of this crossing of the barrier. Okay, so that's the idea, that if you can compute the free energy surface as a function of the reaction coordinate of your choice, then you can get information about both the thermodynamics and the kinetics of the problem. Of course, for the kinetics, there are much more complications, but we can come back to that point later. So we know this, that we need to talk about the free energy. Yes?
There is one question, why the radius of gyration is divided by n to the power 1/3, and what is n?
Right. So in this case, the n is the size of the polymer. So here, for example, there are some representative polymer structure of different length. These are essentially alkane chains, let's say C40, C20, and so on. And here we have used, since we are comparing the free energy surface of different lengths of the polymer, here we have used some kind of a scaling factor, so that for the collapse state, which is spherical, there the size, the effective size should kind of go as to the power 1/3 of the size of the number of particles, right? Because the volume will go as r to the power cube. So essentially, if we kind of scale the overall size by the cube root of the number, then it will all the systems of different sizes will collapse to the same number. So just for comparing the free energy surface of different systems of different sizes we have scaled so that all the values come at the same range. So this is the spherical collapse state where all the values will come here. Otherwise, if I had put absolute RG, obviously longer system will have larger value for the collapse state. So that's the answer.
And quickly, there is one more question: why the blue line curved downward?
Right. Okay. Well, technically, I didn't want to spend much time on this, but this just to show.
Can I suggest one thing, Suman?
Yeah.
You so many questions on this. Please give the reference so that the original article, so that they can figure it out.
Yeah. So yeah, this was actually a JPCB with Professor Biman Bakshi and myself. I'm very bad with the year, but if you can email me, I'll send you the reference. Yeah. So yeah. So and this was about the polymer collapse transition of polymers, hydrophobic polymers in water. So just to quickly give you the answer. So these three systems have three different stability profile. So in one case,
this is Rajorshi.
Right.
Can I say?
Yeah.
I think you're working in poor solvent, and your
Right.
Fourier exponent is 1/3. That's why you have n to the power 1/3, isn't it?
Yeah, exactly. So I didn't want to. Here we are dealing with the hydrophobic polymeric system.
Collapse,
and, yeah. So and water, it's in the poor solvent. So, but yeah, for a simpler answer, the collapse state, if it's spherical, the radius will kind of scale as n to the power 1/3. So I just gave that simpler answer. Yeah. So and also just to answer the other question, these three systems have different inert stability profile. In this case, the collapse state is more stable. As you can see, the collapse state has lower free energy. But in this system, C20, the extended state is more stable. So this just highlights that your free energy surface can directly give you that information that which state is more stable compared to the other. So you can clearly see that there is a crossover as a function of the chain length. Okay. So now that we know that computing the free energy surface is very essential, and we use basically the ideas of statistical mechanics, essentially computer simulation, to get to the free energy surface. And it's just a very quick rudimentary introduction to exactly what kind of theory goes on. Essentially, statistical mechanics tells us that the distribution, the probability distribution of all the microstates is essentially can be obtained from the Boltzmann distribution. So if you know the energy of each of these states, you get some idea of the probability of that state. And any average observable can be computed as this weighted sum over that probability distribution. And finally, what we are most interested in, that this free energy surface as a function of some reaction coordinate is nothing but, it's called a Boltzmann inversion, where it is nothing but minus kbt of ln px, where this px is the probability distribution of that reaction coordinate. So essentially, if you think about it, it's very similar to what we have seen in that thermodynamic expression. So essentially, free energy is related to the probability or population of a state. So when we'll talk about the population of a particular value of a reaction coordinate or certain population of a substate, if you know the probability or the population, the logarithm of that probability will give you the free energy of the state. As simple as that. So this framework kind of tells you how to compute the free energy as a function of any reaction coordinate of your desire. Okay. And now I'll not go into any advanced technique. Earlier, Professor Nishant Nair's talk involved a lot of details about enhanced sampling techniques and how to compute free energies. But today, I'm not going to go into that, but I'll rather focus on the phenomena that we are interested in. But this kind of gives you the crude and direct kind of simplified idea that what the free energy surface is all about. Okay. So with that introduction, let's now delve into the real problem that we are interested in, is about the mechanism of signal transduction. And normally, when we talk about signaling, that will involve this communication between proteins at different levels. So we can think about, for example, there are these three proteins, and let me just turn on my laser pointer. So here we are talking about three proteins. Here there is protein 1, 2, and 3. For example, this protein 1 came up with that information. Now it has to pass on to this yellow protein, protein 2. And secondly, this protein 2 has to pass on this information to protein 3. So essentially, we will now talk about protein-protein interaction, where one protein is interacting with the other protein to pass on this information, and that will also involve molecular recognition. Why recognition is important? It has to recognize its right partner, right? So it cannot give the information to the wrong guy. It has to give it to the right person. And this molecular recognition normally happens through as many of you have probably learned in your textbook, that it requires, first of all, the shape complementarity. It has to match the shape of the right partner. For example, here you can see the shape, the shape at the binding interface is complementary to each other. And also there should be interaction in complementarity. And when we talk about interaction complementarity, many times we talk about electrostatic complementarity, because if you have a negative charge on one protein and positive charge on another protein, you'll have a very strong interaction. So in general, this kind of complementarity is very crucial to establish molecular recognition. Now, this is about inter-protein communication, but there is also intra-protein communication, and that is another term for that is allosteric. So if you have a large protein, or it can be a multi-domain protein, same protein with multiple domains, it may so happen that, let's say, some molecule comes and binds to one side of the protein, and the activity can happen on the other side. So it is, in some sense, biomolecular remote control. So action at a distance. And we'll talk about such phenomena of allostery at a length today. Okay, so these are the two modes. Remember, the one is the inter-protein communication, another could be the intra-protein. And maybe it same can happen in a multi-domain protein. Okay, so now let's take a few examples, simple examples first, to establish how to think about the signaling processes from a molecular thermodynamic point of view that I have just mentioned. So let's take the most kind of a cool example of how do you see, and it's a very fascinating problem that what happens when light shines on your retina, and how then that information is processed in terms of some molecular processes, and then how the process goes to the brain. So we'll not go till the point of brain, but let's first see what happens when the first activation light shines on your retina. So there is this protein called rhodopsin. It's a huge class of protein, but that rhodopsin has a chromophore inside it, which is called retinal, and that retinal has this kind of a conjugated Pauline kind of structure, and depending on the species, for example, for human being, in the native state, this 11, this double bond at the C11, C12, this double bond, as you can see, is in cis conformation, cis configuration. But again, for microbes, microbial rhodopsins, this is the region of interest, this C13, C14, and this exists in a trans configuration. So the native state is all trans for a microbial rhodopsin or retinal, and then for human, this is 11 cis. But something very interesting happens when light shines onto it, and it absorbs light, and then it goes to an excited state. Now, in the excited state, the energy landscape of this retinal is different. For example, in the ground state, this 11 cis was stable, and for this case, the trans was more stable. But this I have borrowed, these diagrams I have borrowed from some other literature, and here it clearly shows in the excited state, there will be a stability crossover. So for example, in the ground state, if this 11 cis is more stable, in the excited state, the trans will become more stable. Similarly, in case of this microbial, this trans is more stable in the ground state, but in the excited state, the cis will be more stable. So as soon as you shine light, the stability changes, and the molecule rotates around that double bond. Okay, so just remember this, that this in the excited or the perturbed state, it has a different energy landscape, and there is a stability crossover. So as soon as now you think about it, that this retinal is actually attached to your protein. So this part, this aldehyde group is actually covalently attached to a lysine in your protein, and when there is a rotation inside that molecule, that rotation is like a bicycle pedal kind of a motion, and it pushes the molecule and it generates a stress inside the protein, and then there will be a subsequent conformational change inside the protein. So first you shine light, there is a stability crossover, retinal rotates, and that rotation leads to a stress, conformational stress on the protein, and there is a subsequent channel opening. So in this rhodopsin, because of the channel opening, it can either pump protons or some ion can pass through, and essentially that is your signal, some kind of a charge going along. But the basic idea is that there is on the perturbation, there is a stability crossover, energy landscape changes. And let's now try to generalize this idea. Okay, so it's not just about vision. So let's think about it, that let's say we can, in the resting state of any protein, the free energy landscape, let's say, looks like this. And this is the conformational coordinate. So we are just plotting free energy as a function of the conformational coordinate. And this is the more stable state. That's where it is there. But now, let's say you activate the protein by some means, and in that activated state, the other conformation becomes more stable. So the relative stability changes. So let's now think about it, that let's say originally this was the most stable state. Sorry, let me just pen. Yeah. So you were here originally in the resting state. Now, as soon as the signal came, you actually kind of excited the system to somewhere here in this conformation. But that is not the global minimum anymore. So now in that excited or the activated state, the system kind of relaxes to a different minimum. So the conformation is changing. Now, when you take away that signal, and then you come back to that other resting state, conformational state, and then again, that is not the global minimum, and it will flow back through this. So this is the general kind of hallmark of any kind of processing of perturbation or signaling. Now that perturbation can be light, it can be binding with ligand, small molecule, any peptide or protein, and also it can be, let's say, change in the protonation state or the pH because of change in the pH. So this is essentially the hallmark of signal propagation or sensing action, that there is a stability crossover or switching on perturbation. So now this activated state, just think of it, this signaling as a second kind of another coordinate. If you think of axis in perpendicular direction, so it's a different another reaction coordinate along the activation profile. Okay, so that can be a ligand binding coordinate, it can be a protonation coordinate, and so on and so forth. But what is actually happening, that in the activated state, there is a stability crossover, and the system essentially, to minimize the free energy, it has to go to the other state, and when you remove the signal, it goes back. So there is all the sensing devices kind of operates through this kind of stability crossover. Okay, so now let's very quickly introduce some languages. These are very rudimentary textbook languages. So there are, when you talk about binding in general, since we are going to talk a lot about binding of ligand and proteins and so on. So there are normally three kind of classes of binding mechanism people talk about. One is that textbook lock and key mechanism kind of thing that you have heard in the context of enzyme catalysis and so on, that the enzyme recognizes its substrate very specifically, and because it's like a lock and key, it exactly matches the shape, and exactly that shape complementarity that I was talking about. So the classical lock and key mechanism invokes the idea of rigid receptor. So as you can look at, we can see here the protein is essentially the conformation of the protein does not change, and the ligand kind of just locks there. So there is no change in the protein conformation. Now this view is obviously is kind of to be discarded at this point because actually protein is not a rigid entity, and the protein is actually a very fluid and flexible entity. It undergoes lots of shape fluctuations. So now there are other two classes of mechanisms that normally people talk about. One is the induced fit. So now in the induced fit, what happens is this particular protein, and let's say now the shape of the ligand is somewhat different. Earlier it had this exactly same shape as the protein active site, but here now the shape is different, the square rectangular shape. But when the ligand comes and binds to the protein, because of the interaction between the protein and the ligand, here there is a change in the shape. As you notice here, the protein conformation changes here. So first the ligand comes and binds to the active site, and then because of the protein ligand interaction, it induces some conformational changes on the protein, and then there is this actual binding. But then there is another possible hypothesis. It's about conformation selection, which says that the protein itself can exist in two possible conformations. As you can see here, the protein, even before the ligand binding, the protein itself can exist in this triangular shape and the square shape. And let's say the square shape is somewhat less populated. Its free energy is higher compared to that rectangular shape. So this state is kind of an activated state of the protein. Just remember this term, that although in the crystal structure we see protein having some well-defined structure, but in reality, when you have protein in solution, it's possible to have some slightly higher free energy states which are low lying and very close to the native state, but essentially the native state can be considered as a collection of many such conformation, but this state will have a lower population as compared to this, but then this conformation will have exact shape complementary to this ligand, and then it will bind. So the basic difference between the induced fit and the conformation selection hypothesis is, in induced fit, the ligand binding is crucial, it happens first, then it induces the conformational change, and in the conformation selection hypothesis says that all these protein conformations already pre-exist. There is this population and ligand only chooses one of them and kind of locks that conformation after it binds. Okay. So now we can put these two pictures together in the same diagram. So now let's introduce two reaction coordinates. One is this conformational coordinate along the y-axis, where you can see that the protein conformation is changing, and in another direction it is the binding coordinate. You might think that if you go from left to right, there's binding going on. So now by using this multiple reaction coordinate, these two extreme mechanisms can be put together within the same framework like this. So if we go along this pathway, that let's say I start with this triangular conformation, then first I go along the binding coordinate in this direction, the ligand comes, and then the conformational change happens. So that clearly is the ideal induced fit pathway. But then if it goes to the other way around, that it's possible that the protein can first change the conformation, it will come down first, and then ligand binding occurs. So that pathway will be conformation selection. So as you can see, by using multiple reaction coordinate, it is possible to compare multiple possible mechanisms within the same framework, and of course the reality could be somewhere in between. So here we have talked about these two extreme possible mechanisms, but there can be somewhere in between, and even more importantly, both pathways could be possible. Let's say in some cases there could be 80% induced fit and 20% conformational selection and so on. So there will be flux along multiple possible pathways, and we can essentially try to understand the kinetics by involving all possible pathways. But this is the framework that how we try to understand the mechanism of any complex biomolecular processes by comparing, by using intelligent choice of these reaction coordinates. And often to understand the mechanism, we need to involve multiple reaction coordinates. Often that is crucial. Now, one question remains, that what is this X? This X I talked about, this reaction conformational coordinate. So here in a schematic diagram, you can say that it's going from square to triangle, but in a real protein, where it is like a dough, which is very flexible, it is just changing its shape, maybe continuously. So how do you even talk about a mathematical quantity that can characterize all this conformational state? And it's not necessary that there will be just two states. There can be many intermediate states. So this is a challenge in itself, that how to characterize mathematically all these conformational states of a protein. Okay, so this kind of sets the stage, the kind of questions that we'll ask in terms of this population shift in the conformational states, and also by choosing this reaction coordinates, how we can think about the underlying mechanism of the process that is happening. Okay, so with that, now let's,
before you move on, there is a question from Sartini Gupta. Can we determine anyhow if the protein is changed after the ligand binds, or the protein already was in the alternative form?
Right, yeah, that's the goal actually. But to properly understand this question, exactly we need to quantify like this, that let's say we can monitor both the conformational change by some parameter X, and then you can also monitor the ligand binding coordinate, and you just see which happens first. If you see, by monitoring both the reaction coordinates, if you see that the binding happens first and then the reaction conformational change, then it's an induced fit. And if you can see that the conformational change happens first and then the binding happens, then this is the other pathway. So yes, we can, and that's the strength of using this molecular dynamic simulation kind of technique. Although we have to actually use enhanced sampling kind of techniques to actually see all those low populated states. But in principle, this is the idea, that by looking at the coupling between two independent reaction coordinates, we can comment on the mechanism. Okay, so now let's talk about inhibitors. So essentially, whenever we take medicines and drugs and so on, they are essentially they will inhibit or rather I should say modulate the action of some enzymes in our body, right? I'm telling this because the next problem that I'm going to talk about is in the context of such inhibitors. So now traditionally, all the medicines that we take, they're mostly competitive inhibitors. What do I mean by that? That in enzyme catalysis, let's say this is the enzyme and this is its natural substrate, this yellow guy. And that, so this is the natural substrates and this is the binding site, and it exactly matches there in binds and it goes to the product. So this is the substrate, then it binds and then the product is released. Okay, but instead, if you put another molecule, this guy, this is the inhibitor, let's say, and this has higher binding affinity as compared to the natural substrate. So obviously, you will get higher population of that inhibitor binding to the active site and not the natural substrate. So now you have kind of blocked the active site by the competitive inhibitor. So it is competing with your natural substrate. So now the competitive inhibitor will bind and the natural substrate will not bind. And hence that bad enzyme, whatever that enzyme was doing, let's say it was the bad job. So we are just blocking that enzyme. So it will not work. Okay. So that is one traditional way of designing drug molecules, that you try to find molecules which will have higher binding affinity as compared to its natural substrate. But we can take another strategy, and that is a more recently people are kind of exploring this strategy, whether we can have allosteric inhibitor. So this term I have just introduced, and let me just quickly explain what do I mean by allosteric inhibitor. So as I said, that this is the natural active site, okay, but it's possible that there is another site somewhere a bit far away from the natural active site where the conformation can change depending on whether your substrate has bound or not. As you can see here, probably here the shape was like a square, let's say, when the ligand was not bound, and here the structure has changed slightly after the ligand has bound, the substrate has bound. So clearly there is some kind of a communication between the ligand binding site and the allosteric site. But now let's say I can design one molecule which can bind to that allosteric site and it can lock to that square-like conformation. It will not allow it to go to this conformation, right? So you can, if we can, let's say, so now I have an allosteric inhibitor which will bind to this site and it has kind of locked this conformation, and then now the natural substrate cannot bind anymore. So essentially, this is essentially allosteric inhibition, that you are designing a molecule that can bind to somewhere else and then inhibit or affect the actual process that is happening somewhere else in that active site. Okay, so this will be our strategy. In the next problem, we'll try to think about ways of designing such allosteric inhibitors. Okay, so this is the problem that first we are going to deal with. So the question will be how to lower your LDL level in your blood. Okay, so of course you know that LDL is the bad cholesterol, and it will be good to lower that bad cholesterol, right? So now
there is a question, Suman. Does shape complementarity somehow influence binding affinity when there is a competition between natural substrate and an inhibitor molecule?
Yes, see, shape complementarity will be always required for binding. So if it is not either the whole, so let's not think in terms of shape complementarity. The point is to be a good inhibitor, it has to, for a competitive inhibitor, it has to have a higher binding affinity as compared to your natural substrate. If it has higher binding affinity, of course, it will compete with the natural substrate and it will not allow the natural substrate to bind. And so it may happen through shape complementarity or electrostatic complementarity. How the binding affinity will be more, that is something that can be controlled. So it can be, it can be even allosteric, as I said. So there can be many ways higher binding affinity can be designed. But the basic principle is that the binding affinity of the inhibitor should be higher compared to the natural substrate. Okay, so now maybe I take some of the questions later after the talk, because there is a chance.
Yes, sure. Let's do that.
Some of the questions, because these are things that will require some discussion, maybe. So let's continue with the flow for the time. Yeah. So So the question is that how do we lower the LDL level of our blood? So now let's see that how LDL is processed. So I'll explain the biology part very briefly, and then we'll go into the chemistry, the actual small molecule part. So what happens is in our hepatocytes, there are these proteins called LDL receptors, and they actually bind to these LDL particles, cholesterol particles. And when LDL binds to these LDLR receptors, essentially they will get absorbed in these hepatocytes, and then they are kind of recycled, and so it will destroy that LDL particle inside the cell, and then the LDLR will be recycled to the hepatocyte surface. So if the more this recycling, essentially the more is the LDL decomposition. So LDLR is going to the surface, trapping LDL, going inside, destroying the LDL, and coming back. So this is the pathway that will have the LDL recycling process going on. But interestingly, there is another regulatory protein, PCSK9, this small guy, this thing. So if that PCSK9 binds to the LDLR, what happens is, so it takes an alternative pathway with this PCSK9 where the whole thing is decomposed in this pathway. So PCSK9, LDL and LDLR, whole thing is decomposed, so the recycling does not happen. So in the presence of, without PCSK9, the LDLR receptor is recycled so that it can continue to consume LDL. But when a PCSK9 will bind to LDLR, the whole complex will get decomposed, so LDLR does not get recycled, so the LDL level will go up. So essentially, PCSK9 kind of will down-regulate the consumption of LDL in the hepatocyte. Okay, so this is known from biochemistry, and it has been found that in several people with mutations. So the idea is that if PCSK9 can strongly bind to LDLR, so if PCSK9 binds to LDLR more strongly, the LDLR does not function, so LDL level goes up. So some mutations which will increase the LDLR PCSK9 binding affinity will lead to higher LDL level. But if some mutations actually reduces that binding affinity, it will lower the LDL level. And there are such genetic modifications where it has been found this to be the case. So this could be a strategy that if somehow we could lower this, suppress this, essentially this PCSK9 LDLR interaction will suppress the recycling of LDLR. So what our objective is to block this PCSK9 LDLR interaction, and hopefully allosterically. Okay, so this is the idea that we would like to reduce the binding affinity between PCSK9 and LDLR by adding some small molecule, hopefully allosterically, and let's try to see that what could be the possible strategy for doing that.
Suman, sorry,
Yeah.
This is Shravani here. So why do you specifically say allosterically?
That's our goal. So, well, okay, the correct answer is actually the PCSK9 LDLR binding surface is rather large. So technically, having a competitive inhibitor is not easy. So there are other strategies for designing antibodies. So then you need a whole protein in order to block the PCSK9 from binding from LDLR. So since the binding surface between PCSK9 and LDLR is large, having a small molecule inhibitor as a competitive inhibitor will be difficult. So our strategy is hopefully that we'll find a small molecule to bind somewhere else that will reduce the binding affinity between PCSK9 and LDLR. So this is our end goal.
Okay.
So let's see whether we succeed there or not, and what would be our strategy. So this is like the structure. We are not dealing with the full LDLR, but only one domain called EGFA. So this blue guy is one LDLR domain, EGFA, that is binding to the PCSK9, and this is one part of the PCSK9 protein. Now, interestingly, there are some loops in this PCSK9 protein, and these two loops we'll focus on. We have found that this particular loop can exist in multiple conformation. Okay, so well, technically these loops are not resolved in crystallography, but we had to model these loops. But we have actually started with multiple possible loop conformation, and then we kind of discovered that actually there are distinct loop conformations which are significantly stable. So essentially there are significant conformational heterogeneity or plasticity in these disordered loop regions. And here we schematically show that these loops, how they can exist in multiple conformation, although they are technically disordered. But all of this in this conformational space, what we show that each are different trajectories and long enough, 1 microsecond long trajectory. And you can see that in the loop tends to get stuck in some local minima and it stays there. Okay. So it's not like the loop is very flexible and it is visiting all the conformational states very fluently, but it can get stuck in distinct conformational states. So then we ask this question, whether there is any correlation between the conformational states of these loops and the binding affinity with this LDLR domain. Okay, so that is the hypothesis, that if what we have noticed that depending on whether it is bound to this EGFA or not, the conformational state of the loops can get shifted. So there will be population shift in the loop conformation depending on whether you keep the ligand there or not. So our hypothesis is it could be the other way around, that this depending on where you keep this loop, in which conformation, the binding affinity of this LDLR can be different. And indeed, that seems to be the case. So, okay, so this is the question, that since ligand binding seems to modulate this loop conformation, can it be utilized the other way around? That if we tune the loop conformation and the binding affinity in turn gets modulated. Okay, so let's talk about some basic ideas about identifying allosteric drugs. So as we talked about, that we need to find such sites, and these are often called cryptic sites because those sites may not be open in a normal crystal structure, because those are low populated states of the protein conformation. And also we'll talk about this reversibility of the conformational thermodynamics, and this point I'm going to come back to. So the basic idea is that depending on the binding status, that whether the PCSK9 is bound to the LDLR or not, that allosteric site can exist in two different conformations. And now if I do it other way around, if I lock it to one specific conformation, I can reversibly control what will be the binding affinity of that particular substrate. So here, using this basic thermodynamic cycle, I try to establish this idea that imagine that this protein has two possible binding sites. This is the natural binding site, where this is the ligand, and this is the free energy of binding, delta Gp. Okay. So now so this is the free energy of binding for natural substrate when the allosteric site is in this square conformation. But now imagine that there could be conformational change in this allosteric site, where it goes from the square conformation to the triangular conformation. And similarly, in the presence of the ligand also, there could be conformational change from the square to the triangular. Okay. And so now we are talking about two kinds of binding affinity. One, when the allosteric site is in the square conformation, this is the binding affinity of delta Gb. And when the allosteric site is in the triangular conformation, we are talking about the binding affinity of delta Gb dash. So essentially, this kind of summarizes the idea of allosteric control of binding affinity, that depending on the conformation of a distal site, the binding affinity is getting changed. But now we can kind of close this thermodynamic cycle and show that this binding affinity will be lower, that is less negative, that delta Gb dash will be higher if this conformational change is less favorable compared to this. So essentially, to summarize, without going through all the maths, to summarize, the conformation that is more favored without the ligand, if we can lock in that conformation, then the binding affinity will be lower. So essentially, we would like to lock the protein in this particular conformation, this allosteric site. If I can lock in that conformation, I expect the binding affinity to be lower. So indeed, that seems to be the case. We have kind of characterized the full free energy surface here. Here in this free energy surface, what you see here, these are essentially different conformational states of the loop, these two loops that I was talking about. So, and as you can see, different here, all these numbers, they mark the binding affinity of between the PCSK9 and LDLR, the binding affinity values for different conformations of the protein. So as you can see, there are some minima where the binding affinity is very large negative number, so its binding affinity is higher, -85. But on the other hand, for this minima 1, binding energy is -41. So here we could establish that depending on the protein conformation, or rather the conformation of the loops, the binding affinity with the LDLR can be controlled. Okay, so that's the basic idea, that we have identified distinct conformational states of these loop regions, and they essentially have different binding affinity. So it gives us some control over choosing that what to target. Okay, and also just to quickly show that what kind of controls these different distinct conformational states, we could find that the hydrogen bonding pattern is distinctly different between these two conformational states. So between we are showing here two states, the state 1 and state 2, and in one case you see there are lots of intra-protein hydrogen bonded configuration, but in other case there are some inter-loop configuration. But all of these conformations are stabilized by unique set of hydrogen bonds. Okay, so this is something very crucial, this idea, this network of hydrogen bond stabilizing different population of conformation. Remember this aspect, and I'll keep on highlighting this aspect in the subsequent stories. So I just wanted to point out that these distinct conformations are getting controlled by a certain set of hydrogen bonded network. Okay, so again here in this diagram we summarize that different conformations, different loop conformation has different distribution of the PCSK9 LDLR binding energy, as you can see here. So essentially we have now a proof of concept, and then we have initial some states of this allosteric, we have kind of performed some virtual screening, and we have identified some allosteric inhibitors which will kind of lock those loop conformations. So we have targeted those loop conformation which has lower binding affinity of between PCSK9 and LDLR. So the idea is if we can lock those loop conformation, then possibly those will be the allosteric inhibitors. And at least our preliminary results seems to be quite encouraging, that with this one sample ligand, we see that after doing MD simulation, with that trajectory, on the ligand bound trajectory, the binding affinity is much lower, so lower negative numbers, much lower as compared to the natural substrate in different conformational states. So essentially, this is our design strategy. So we'll identify that which loop conformation has the lower binding energy, and we'll use them as drug targets, and then we'll try to lock those conformations using this kind of inhibitors. Okay, so that's the simple idea, and I just at this point I would like to highlight this kind of at a conceptual level, that the loops and the linkers may appear to be unimportant in some cases, although of course those who know really biochemistry, they will not agree to this statement. But sometimes by protein structure we mean alpha helix, beta sheet, and so on. But many times this protein, the disordered loops and linkers play a very important role in protein function. And even though they can be deceptively structureless, although they are not structured, it looks like disordered loop, but actually they can exist in very well-defined distinct structural states. So a loop, although it looks disordered, but there are distinct states and very well defined by those kind of hydrogen bonded network. And each of those states can play a very important role in protein function, and often they can be very useful potential drug targets. And you can also think about it in the context of IDPs, because you know that in many signaling pathways, the IDPs plays a very important role, intrinsically disordered proteins, because they are very adaptive. Depending on the environment, they can adopt a shape or a structure, a conformation, and so on. So very similar. The loops also, although it's disordered, you can think of them like an IDP inside a protein, and they can have very distinct conformational states which are biologically important. Okay. Now I'll change gear and go to the next topic very quickly. And so here you see that I talked about something called dynamic allostery, right? So and also in these review articles they talked about that these linkers are not merely flexible. And since I mentioned about dynamic allostery, and very quickly I'll go through the next topic. I don't want to really spend much time on this particular topic because I have talked about this many times before, but I'll just give you a kind of an overview of the ideas behind this problem. Okay, so what do I mean by dynamic allostery? So far, what you have seen in that last example in allostery, that there were some structural changes in the loop conformation depending on whether there is ligand binding or not, or in the other way around, that if you lock the loop conformation to one of them, the binding affinity can change allosterically. But there are some examples, and increasingly more number of examples, where allosteric can happen without any well-defined structural change per se. That if you compare the crystal structures, you may not see any structural change. But interestingly, there will be change in the dynamics of the protein in terms of the side chain motion. So this is this particular protein called PDZ domain protein is a hallmark classical example of dynamic allostery, where this ligand binding to one side of the protein. So for example, in this here, what I show is superposition of two structures with and without ligand, and this is the ligand binding site. And as you can see, there is almost no structural change, but both the solution NMR and computer simulation has indicated that there will be in the distal part of the protein, the side chain dynamics gets modulated. Okay, and it has been suggested that this dynamic allostery is purely entropic in origin and so on. But the first thing to notice is that the static structural view is not enough to understand this kind of phenomena. So you have to invoke the idea of dynamics and possibly this idea of conformational population shift. Okay, so the first question that we ask here, which is extremely important, is whether really there is no structural change, even when people say that allostery without structural change, or we just don't see it. So we need to probably change the way we look for structural changes. So when you look into just the backbone view, this is the traditional view. When you superimpose the backbone, there may not be any structural change. When you look into the residue-residue contact map based on these distances, there may not be significant change, because there could be very subtle rearrangement in the protein structure. So what do we do? So what we suggested earlier in our work, that the interaction energy of the protein can provide a very powerful and sensitive descriptor of the subtle conformational changes or subtle changes in the population in the system. So generally what we do is we compute the average non-bonded interaction energies on a per residue basis and compare them between the bound and the unbound state, or in general perturbed and unperturbed state. So here, for example, delta EI is the change in the average interaction energy of each residue between the bound and unbound state. So without going into details, what I'll just summarize is what we find, that there are a lot of cancellations going on. So here, what I show here, that how for each amino acid residue, how the average interaction energy changes. As you can see, there are lots of large positive numbers and lots of large negative numbers. So there is a lot of cancellation going on at a local level. So although as a whole in the protein, the average enthalpy is not changing much, there are lots of, at a local level, a lot of rearrangement is going on. And that seems to be very general phenomena of such signaling proteins, as I'll again repeat this idea. Now, how this rearrangement, energetic redistribution and rearrangement is happening in terms of this kind of a network. And here we have again followed the residue pairwise interaction, and we have monitored that how the energetic perturbation is propagating from the ligand binding site to the distant site. So here essentially what we are monitoring is the pairwise interaction energy average between ith and jth residue, delta EIJ, and then we are monitoring that which pairs are getting perturbed the most. And if we monitor that flow of energy in that way, we can kind of construct the network, that how the energetic perturbation is propagating from one region of the protein to the other. And here you can clearly see that there is one domain here, there is another domain on this part, connected by this kind of connections. Okay, and how and why this connection is established? Again, this is in terms of this hydrogen bonded network. And what we find is this, in case of the unbound state, there is one set of hydrogen bonds. As you can see that these pairs are kind of, there are salt bridges and hydrogen bonds. One set of hydrogen bond is forming a network. And in case of the bound, there is another set of hydrogen bond. So you can imagine that we can talk about now a population of conformation in terms of a certain set of hydrogen bonds. You can think of it like again a contact map, but in terms of that which pairs are connected to each other. So now you can describe a conformational state of a protein in terms of a set of hydrogen bonds or pairs of hydrogen bond, and signaling will mean a population shift among this hydrogen bonded states, a set of population of hydrogen bond shifting from one set to the other. So essentially, the perturbation due to ligand is getting communicated through this rewiring of this hydrogen bonded network. Okay, so I'll just summarize this idea that essentially what we're talking about is this population shift in this pairwise interaction network, and even a small change in these populations can drive a large change in the overall interaction energy. So in a short summary, this dynamic allostery, if we have to redefine, so all the dynamic changes are kind of driven by the modulation in the underlying energy landscape or in the interactions. So although traditionally dynamic allostery was thought to be purely entropically driven, and it was suggested that there is no change in the total enthalpy or the total energy of the system, what we showed that at a local, although the total energy may not be changing, but at a local level, there is a lot of internal cancellation going on, plus changes and minus changes. And those changes are essentially driven by this rewiring of this side chain interaction network, this hydrogen bonded network. And essentially, there is this population shift of this ensemble of interactions defined by this set of hydrogen bonds. Okay, so this is the idea that we proposed for the first time. And then subsequently, we asked that for the same protein, so far we have talked about one kind of perturbation, which is ligand binding. Okay, so what happens if instead of ligand binding, there is a different kind of perturbation? Same protein, I just, instead of putting a ligand, I change something else. So I change the pH of the solution and I protonate a residue. Can the same protein respond maybe in a different way or same way for a different kind of perturbation? So that's the kind of, we had that kind of curiosity. So what we did is we kind of protonated these histidines here and here independently, and we checked that whether this protonation can also induce some similar kind of allosteric response in distal parts of the protein. Okay, and interestingly, I'll just give you the story. Interestingly, what we find that yes, the change in the protonation also can elicit same kind of response. Same by the same, I mean that same set of hydrogen bonds, that same network is getting perturbed, maybe by different extent. It's not that exactly the response is exactly the same, but the same network is responding to a different perturbation. Okay, and also here you can see one particular example. For example, if you monitor one particular salt bridge, this 305 and 309, let's say this particular salt bridge, which had in the apo form, the salt bridge has a population of 74%, and in the bound form, it was 22%. But in the protonated state, it has actually decreased. It is going in the same direction in the ligand binding. But here, the protonation change is happening here, and the salt bridge is here. So, kind of, even a distant protonation is kind of eliciting the same kind of allosteric response that would have been that would have happened by the ligand binding. So, our hypothesis or our proposal is kind of, this is as a hypothesis at this stage, that there is probably a common set of residues. There's a network built of those residues. It's a universal set of network that can respond to different kinds of perturbation, be it ligand binding, be it can be a protonation change. So the protein might have evolved to develop that kind of a response system that can respond to different kind of perturbation. So this is the hypothesis at this point, that this hydrogen bonded network creates an inherent response system in signaling proteins that may respond to different kinds of perturbation. So remember this idea, and I'll again revisit this in the last next problem that I'll just very quickly I'll summarize. Okay, so far we have introduced a few ideas in, just to repeat this, in this dynamic allostery, we have introduced a couple of ideas that even though there may not be appreciable structural change, visible structural change, there is subtle population shift in the conformation defined by this set of hydrogen bonded network. And then we have also shown that there could be an inherent response system inbuilt in these proteins that can respond to different kinds of perturbation. Okay, so with that knowledge, let's move on to the last problem that I'll talk about. Maybe I'll finish in another 10 minutes or so. Okay, so This is the last problem that we are going to talk about, and this is the probably most fascinating and complex last problem that I would like to talk about. So again, as I mentioned that in cellular machinery, there is a lot of complicated communication is going on, and often there will be multiple proteins involved in a particular biochemical pathway, right? And here, let's take an example of one particular pathway, such pathway. There is this guy, there is one class of protein called RAS proteins, and let's take one subclass of that, which is called a Rho protein. Let's not worry about the names so much. I don't worry about biology in so much detail, but let's just look at the fascinating mechanism that is going on, what the machinery itself. So this protein, Rho GTPase, it is activated when it is bound to GTP, right? And then when it's like a switch, it's a molecular switch. When it is bound to GTP, it's in the on state, and then it will activate this downstream other effectors protein when it is in the on state. Now, when it is bound to GDP, when it gets hydrolyzed, GTP gets hydrolyzed into GDP, and then it's in the off state. And this process is catalyzed by another protein called GAP. And in the reverse process is also catalyzed by another protein called GEF. So you see that even in this small thing, there are already three proteins involved, and there are multiple proteins which are kind of regulating all these processes. We are not interested in this part, but just notice that even in the off state, this protein is anchored in the membrane. So there is this one hydrophobic tail. It's a prenyl moiety or general group. It's just a hydrophobic chain through which this complex is anchored inside the membrane. Now, there's another guy comes in, another protein called Rho GDI. So when this Rho GDI comes and binds to this Rho GDP, what happens is this hydrophobic anchor is pulled out, and that goes inside this in this hydrophobic cavity of Rho GDI. So now you see that hydrophobic tail, when it is trapped inside, this whole complex is solubilized in the cytosol, and that is it's extracted. So basically, the job of Rho GDI is to extract the off state Rho GDP, and it blocks the Rho GDP from being recycled. So it is kind of deactivates the Rho proteins, and it's a dissociation inhibitor essentially. It blocks the GDP from dissociating from the Rho GDP. So but the basic idea is that when it binds to this complex, it will extract from the membrane and it will solubilize it in the cytosol. But also it has to return it back, right? So first it binds and brings it to the cytosol, but somehow it has to go back to the protein when it has to be released. But interestingly, this Rho GDI can actually extract different classes of different subclasses of the same protein, like this Rho, RAC, CDC42. These are three different subclasses of the same RAS group of proteins. Okay. So, but now the mechanism of release of this is the phosphorylation of this Rho GDI. Phosphorylation is nothing but attachment of a phosphate group, essentially a negative charge. So when some other protein like kinase or something will phosphorylate this Rho GDI, it will release its partner. But the interesting question is, it has to selectively release one partner. So because it can be bound to either Rho or CDC42 or RAC. It can be in complex with all these three partners. But interestingly, it has the ability to release only one partner specifically. So there is some kind of a selectivity. And that selectivity originates from something called a phosphorylation code. So let me just briefly explain what do I mean by this phosphorylation code. So here is this here is this Rho protein, and here is this GDI complex. Okay, and here this is this long or rather large binding surface area, and here there is one regulator yarn. But let me just highlight a few of the structural features. So this is that hydrophobic tail I was talking about, this green this tail, and you can see that this is the general moiety, which is kind of buried inside the hydrophobic cavity of GDI. So this is the whole complex. Now there are different phosphorylation sites, and interestingly, biochemistry indicates that depending on where you phosphorylate, if you phosphorylate this serine 34, the Rho A will be released, or if it is serine 96. On the other hand, if 101 or 174, which are here, if these serines are phosphorylated, RAC1 will be released. So think about it, that this GDI is the same, rho GDI is the same, and this is the partner, GTPS. This partner can be any of these three, RAC1, Rho A, and CTC42. Now, depending on where you phosphorylate this GDI, it will release one of the partners. So if you phosphorylate at the wrong site, for the other protein, it will not release, right? So then, like our question, the kind of question that we ask is, how does a specific Rho GTPase will get released from the Rho GDI selectively? So now, that's the statement of the problem, that you have to specifically phosphorylate the right serine residue in order to release the right partner selectively. So we'd like to understand what is the mechanism of this process. Okay. So obviously there is some kind of a long range effect. Again, we'll be talking about allosteric. So if you phosphorylate this serine residues, here it has to detach up to this point. This HTH helix turn helix domain, or the regulatory arm has to move out, right? So there has to be a very long range allosteric of about 3.5 nanometer. And but essentially a phosphorylation means addition of an extra negative charge. So again, we'll speculate that there should be some kind of a rearrangement in terms of that electrostatic interactions and hopefully hydrogen bonded network. And let's see. So I'll just kind of quickly go, because there will be a lot of details as you can see, but I'll just quickly gloss over the details and just tell you the moral of the story in a nutshell. So what we observe is that after we phosphorylate, indeed, lots of things start to happen. So what we expect is, on phosphorylation, the protein should detach. And now for the detachment, several things should happen. One is, it has to pull out this geranyl group, because it is buried inside the hydrophobic cavity, it should come out. Another thing should happen is this helix turn helix domain, it has to detach also, right? So several things should start to fall apart if you phosphorylate, right? So indeed, that's what we see. And for example, the first step is the retraction of the geranyl tail. So this tail has to come out. And what we find that, again, for that retraction process is associated with breaking of some salt bridges between this polybasic region, PBR, with the cavity. So these interactions will break and some new interactions will form on this other side. Okay. So essentially, let's try to highlight this few specific hydrogen bonds. So for example, if these hydrogen bonds break, they will indicate that this, if you imagine that there are some rubber bands, some elastic bands attached between this PBR and the cavity, these are breaking. And on the other hand, these hydrogen bonds are forming. So it's kind of pulling out in the outward direction. So again, some breaking of some specific salt bridges and formation of another set of salt bridges will effectively lead to this outward pulling motion of this geranyl group. Okay. So that is one mechanistic picture, that how certain mechanical motion can be reproduced by this breakage and formation of certain specific hydrogen bonds. Another example, that this HTH domain has to kind of be pulled down. It has to be pulled out, right? So there has to be some kind of a drawbridge motion that we actually notice that on phosphorylation, this outward motion of this HTH domain increases. So it's kind of the flexibility increases and it comes out. But again, what causes this pulling effect? So if you kind of imagine that if you have to pull down something, there has to be some kind of a pulley, and you have to pull some strings, there will be some cycles and so on, right? So even for a drawbridge, we need some pulleys and some strings. So similarly, we have identified that again, there are certain specific interactions. For example, these pairs of interactions, if you think that if you contract them, if you form those interactions, you make them stronger, and then that is contracts, and that effectively leads to some kind of a pulling motion. So again, on phosphorylation, these salt bridges become more stable. That leads to this kind of a downward, outward pulling motion. So what we could establish is that several such, first of all, on phosphorylation, there is a cascading effect of these salt bridges and hydrogen bonds rearranging with each other. And then that leads to this kind of mechanical motion that corresponds to this detachment or pulling out of this from this between these two proteins. Okay. So I'll just summarize at this point. I'll not go any further. We have also looked into, so as I said, whatever we have studied here so far is this particular phosphorylation, this serine 101174, and we could show that in case of RAC1, actually it leads to weakening of interaction. But subsequently, we have also looked into this other protein called Rho-A, and we have shown that in case of this particular phosphorylation of serine 34, there is weakening of interaction. But instead, if you phosphorylate the wrong set of residues, the weakening does not happen. So actually, we could in our simulation, we could actually reproduce the correct trend of the binding affinity, although this is at the level of MMPBSA binding energy. So here it could show that compared to the wild type binding energy distribution, in the correct phosphorylation sp34, there is a significant weakening of the binding interaction. But if you do the wrong phosphorylation, the binding energy actually even increases slightly. So at least the correct experimental results are reproduced in the effect of end result of the phosphorylation. And I'll not go through the further details, but I just wanted to highlight this aspect, that what we found again, that both you can see that here I have colored by color, I have highlighted which regions of the proteins gets part up energetically. Okay, so this for the this 34 phosphorylation, this is where the perturbation starts, and this is the allosteric pathway. Along this pathway, the energetic perturbation gets propagated, right? Because this is where the geranyl tail is there. So we can see that again, that energetic rearrangement is giving you this allosteric propagation pathway. But more interestingly, even for the wrong phosphorylation, you see that the phosphorylation was somewhere here. Then also there is a kind of energetic perturbation along the same pathway, right? So again, I would like to highlight our earlier hypothesis that there could be a same set of network residues which are probably leading to the response, right? So what I'm trying to say, even though here the change in the binding affinity is not as much because this is not the correct phosphorylation, but again, this perturbation also leads to some weak energetic perturbation to a lower extent, but still the same set of residues along the same pathway, you can see that the perturbation is propagated. So I'd like to just highlight this universality kind of thing, that probably there is one universal pathway involving a certain set of residues that is kind of helping the allosteric propagation, irrespective of the specific nature of the perturbation. Okay, so at this point, with these ideas, I would like to just summarize. We have actually looked into the energetic propagation pathway in detail, and again, the same idea of hydrogen bond rearrangement that is leading rise to, again, this allosteric response. But again, these are too much of details. I have already told all the story that I was supposed to tell. Okay, so at this point, I'll just say that this is kind of future. We are going ahead. So we would like to kind of build a Markov state model kind of to describe the kinetics of the problem, where the Markov state model is nothing but like a chemical kinetic kind of a picture, where there is transition between various substates. And if we know the kinetics of these individual states, then we can kind of construct a full kinetic model. And if you want to know about more about Markov state model, I think one of our experts in this group, I don't know whether Jagannath is here, he's one of the experts in Markov state models, and he can talk about it in a future meeting. So what we are trying to do is to build, so since we have talked about this kind of conformational states in terms of this certain population of hydrogen bonded network, and now we would like to see that how one network, what is the kinetics of the transition between one set of conformational population to another, and whether we can build an overall kinetic model out of these transitions between the different set of conformational states. Okay, so with that, I would just like to summarize. I'll skip this. So just the moral of the story is the biomolecular signaling is all about an intricate interplay of various kinds of interactions and of the modulation in the underlying conformational landscape. Okay, and there are different kinds of interactions, of course, although I have primarily kept talking about electrostatics and hydrogen bonding, but of course there are other interactions, hydrophobic, van der Waals, solvation, and so on. And most important thing is that that interplay is placed such that the triggers, there are these external triggers can reversibly tune this underlying energy landscape so that one population can reversibly shift to another population, it can also come back. And also an ensemble view is very crucial. So where we can talk about this different conformational states as a population and their interchange between all these conformational states. Okay, so I think that's all for today. And I must thank the people who have contributed to this work. The first work on PCSK9 was done by Dr. Ipshita Basu. She's a postdoc in my group. And then Amit did the work on the dynamic allostery and also the RAC1 rho-GDI interaction. He has left the group now, and further the work on rho is being continued by Krishniti. Okay, and I thank for the funding ACRB and SNBNCBS Center for Computational Facility. Thank you all for your kind attention, and I'll be happy to take any questions. And also for the students, if you have any questions, if you need any references, please feel free to write to me. Many of the information I don't remember offhand, but I'll be more than happy to communicate with you at a later time. So thank you.
Thank you, Suman, for the fascinating talk, and really have expanded this whole layer of allostery, which started with your PNS paper. So I will start asking questions from where we left while you were speaking. So the question was, Ayan Ghosh asked is there any advantage of using allosteric inhibition over competitive inhibition? And I guess you have already answered this, right? That where the main site cannot be
right.
Yeah.
So actually, right. So there are one clear benefit is that when the main site is not accessible or it can be too large, it might be difficult to design competitive inhibitors. Also, an allosteric, and it has been suggested, although I don't know exactly why, but it seems to be that the allosteric modulators can have somewhat lower side effects because they will be more specific, or it will be more, there is a higher degree of control. So it does not affect too many of other biochemical pathways. So it seems that the toxicity effects will be lower for the allosteric inhibitors. But primarily, yeah, so many a times it is not possible to design a good competitive inhibitors.
So then this question is by Satish Dasari. What is the collective variable used to sample the loop free energy?
Right. It's a very good question. In this case, it's very complicated to describe the loop conformation actually to come up with a good CV for a disordered loop. And actually, maybe I'll just go back to the slide. So normally a better ideal way of doing it is to define some kind of a path variable that is used in metadynamics community, where you can kind of define a initial state and the final state, and then kind of linearly extrapolate, and then it's similar to large elastic band approach, and then you find some intermediate milestones along that pathway, and you kind of compute the distance from along that pathway that connects from one state to the other. In our case, we have simply used RMSD as the reaction coordinate, but we have used two different RMSD. One with respect to the reference, maybe I'll just go back to the slide. Yeah, so in this slide, as you can see here, so we have two different RMSD here. Cluster 1, so here we have two different references. Let's say I have two extreme structures. So if I take two different RMSD value, one with reference to one cluster, one structure, another RMSD with respect to another structure. So in this space, conformational space, we have used these two different RMSDs. So if you see that here, if the RMSD is low for cluster 2, it is more similar to this, so you will be somewhere here. And if for your cluster 1, you will be somewhere here. So this space defined by these two RMSD values kind of quantifies the whole conformational space. And in the free energy surface also, we have used these two reaction coordinates, essentially RMSD from two end states, two extremely different crystal structures. This is a bit delayed. My slide I'm changing, but it's coming a bit late. So, but yeah, so here along x-axis, this is RMSD with respect to one conformation, and y-axis is RMSD with respect to another extreme conformation.
So in this figure, don't you have something just up there which is even lower minima than, means I thought the 4 of there, you haven't marked the first one, which is the tail of, like, which is the beginning, the first one, the yellow.
Yeah, so those are, I mean, actually this free energy surface is still a bit preliminary, it might not have converged, but yeah, and also this this is not may not be a significant population, but we have not explored that. So it's possible that that conformation may have even lower binding energy. That is possible.
Right, it appears so.
Yeah. So actually what we have done is we have taken these trajectories and we have kind of performed clustering and took the most populated clusters, because actually even to target that particular conformation and to dock a ligand, that structure should have some well-defined, I mean, some appreciable stability. But if that state is not too stable, I mean, not, I mean, it's too unstable, then it may not be a good target also.
Okay. So the next question is from Ratin. How to select the representative points to explain the flow of energy? I think it's all started with your allostery thing where you talked about hydrogen bond breaking and reforming. I guess that's when the question was asked, but Ratin, you can unmute yourself and ask the question what exactly you mean.
Hello, sir. Am I audible?
Yes, please go ahead.
Yes, you're audible.
Hello. Sir, yes, sir. I was talking about these residue pairs.
Right.
How to select it? Do you need to select it by visual inspection or?
Right. So no, actually we are using kind of this interaction energy to shortlist that which could be important. So exactly what we have done is we have computed all the energy, average energy between all pairs. So all ij pairs, if you compute the average pairwise interaction, non-bonded only, average non-bonded interaction energy between all pairs, and then we notice which pairs has changed the most. So here we have these numbers of delta Eij for all pairs, but we only visualize that which delta Eij is more than certain cutoff value. As you can see here, that certain lines will indicate whether it is more than 3 kcal per mole or 10 kcal per mole or so. So we kind of see that which pairs have changed the most. So that means that those pairs have been part of the most. And then we subsequently we look into try to understand why those pairs have changed the most. And we notice that those hydrogen bonds or salt bridges are undergoing the largest rearrangement.
So I'll move on because there are many more questions. Abhinandan Das, I don't know if he's there, but he asked how to decide which specific residue pairs to select to calculate residue.
This has been already answered. We calculate all the pairs.
Yeah, exactly. Then there is some comment by Anand, which we'll read later. Maybe let's move on to Tarak Karmakar. He said he wants to ask a couple of questions. So Tarak, you can unmute yourself and ask whatever you want to ask, but maybe keep it short.
Thanks. I'll try to take only a few minutes. Thank you, Sumanda. It's always a pleasure to listen to you.
Thank you.
Fantastic talk. And I have many questions, but I'll ask you in person.
We can always discuss later. Sure.
Yeah. But one thing that I was wondering, can water kind of induce allostery? I guess it can, right? Water molecules.
Well, I don't know whether I should say it induces allostery, but it definitely plays a role in the thermodynamics of the allostery. So what I mean to say that when I talked about, let's say in this case, let's say ligand binds here and there is a change in the dynamics on this side, right? But now, actually even the water dynamics will get modulated on this side. Because let's say, because of the rearrangement in the protein structure, some, let's say, charge residues has opened up. So the solvation structure around those regions will change. Sure. So obviously the solvation will also dictate that how the protein conformation will be changing in that region. So the role of water is kind of inbuilt inside water will dictate that how the underlying conformational free energy landscape will be induced. And also there are some examples like in GPCRs, for example, often people find that there are this hydrogen bonded chain of water molecules, and in those cases, even this rearrangement of hydrogen bond can propagate through the hydrogen bonded network of water. So that could be one example of purely water. I don't know, people don't say it is allostery, but when, let's say, some perturbation is propagating through the hydrogen bonded network of water, you can call it allostery, possibly. But definitely, even for proteins, the solvation effects will implicitly control the allosteric effects.
That's true, but I was thinking that if there are some trapped water molecules at some pocket, that kind of can mediate the allostery.
It's definitely possible. See, the way we think about it, for example, here we have kind of, in this case, there is no trapped pocket in this particular example. For example, here we have kind of come up with this hydrogen bonded network, right? Now, it may so happen that in those cases, when I will look into this hydrogen bonded network, those trapped waters will participate in this hydrogen bonded network, because many a times there will be water molecule bridging. There are bridged water molecules which will like hold hands between two amino acids, right? So water can actually mediate the interaction between two different distant parts of the protein. There are such examples. Yes, those can be important. But in my test cases, there are no such bridged water molecules.
Can I ask another quick one?
Yes. Quick.
Okay. So I was wondering, so you talked about the dynamic allostery, and there's another one, electrostatic, or am I missing? Or
I kind of argued that the dynamic allostery is driven by electrostatics, but the main idea of dynamic allostery traditionally was where there is no structural change, but there will be the change in the side chain dynamics even in the distal part of the protein. So the term dynamic allostery was coined to talk about where there is no structural change.
So on that line, so I was thinking that if there is an interplay between the entropy and enthalpy.
Exactly. So that has been the first of all, that the traditional view has been that the enthalpy does not play any role, only entropy. So obviously entropy plays the most important role, but what we wanted to actually highlight that just because the total enthalpy does not change, that does not mean that it does not play a role. What I wanted to show that because of this internal cancellation, that one set of hydrogen bonds is going into another set, there is cancellation, but then that is what is causing the energy to be redistributed, and that redistribution of energy is essentially related to the entropic change, right? But we wanted to show that the underlying effect actually happens through the rearrangement of this hydrogen bonding network. So it is not true that enthalpy does not play a role. So we actually question that view. But of course, entropy plays a role, and there is enthalpy entropy compensation as well.
I see. Okay. Okay.
Thank you. And also you found this network to be constant for a particular protein, right? That is the other concept that you want to.
Yes, the network is kind of universal. It is preserved.
Universal,
the specific response will depend on the kind of perturbation, but the same universal set of agents respond to that different perturbation.
Yeah. So the next is from Nilanjana. She said she has a question, rather a comment, and she wants to say it. Yeah.
Okay. So thank you, Sarika. Suman, wonderfully lucid talk as expected, and as we have heard, thank you so much. There are plenty of students from ICER as well, I think. I actually had a, more of a curiosity is, as you have so elegantly shown that, this redistribution of electrostatics underlies the allostery in the proteins that you have investigated. However, to my mind, there are some well-established models such as this Munoz-Itten model, right? Which kind of argue that, allostery is more of a stochastic diffusion in an energy landscape. So I was wondering if that view can somehow be reconciled with your new findings, or, have there been questions even from referees on that, or, that view and this, what you have just come up with appear to me, a little bit contradictory to say the least. So I was wondering if there's a natural reconciliation of these two points of views.
Well, I am not aware of the work that says it is purely stochastic diffusion, because in that case,
model, this Munoz-Itten WSMA.
Maybe you can share that literature with me, because my argument would be if it is a purely stochastic diffusion, it will be difficult to control, right? So nature would like to have a very tightly gripped control over how it will respond to certain perturbation, right? So it cannot be completely stochastic and random. So
No,
what
I
I
agree with you. Yeah. And, this model has been around for more than 4 decades, I think, but it's still being used. We get a lot of literature where that's kind of taken as a gold standard and, people look at allostery that way.
Yeah. And
So,
another
but-
alternative, what I may mention here, that there are now, how do we look for allosteric pathway? There are many different methods, and I did not have time to discuss all those. Mostly people look for correlations, for example, that you look at protein dynamics, and that's a standard way that you look for whether certain part of the protein is correlated to the other part. So we don't take that route. So I am more, because it may so happen that in one energy basin, certain regions is correlated, but it can go to another basin there, the nature of correlation can change. So I'm more interested in, hello?
I just want to add something because it's the discussions on stochastic diffusion and things like that. Actually, if you go back to Feynman's thing and try to map it a little bit into evolution, evolution is always a very driven process. There is not much stochasticity, as Suman was correctly saying. What actually happening, there many small things are adding together, and that means 1 + 1 is not 2 in the biology world. When actually 1.1 is more than 2. That because they reinforce each other. So many of these processes have been evolved to this chain of hydrogen bonding, because when 2 hydrogen bonds forms, the third one becomes easy, and those two become stronger. Since the reinforcement is one of the microscopic or molecular level of the biology of evolution, which actually one can look into a little bit more carefully.
Right.
That why
Actually,
one
this exactly what you said, that this cooperative effect is actually the one hallmark of allostery, like this classic example of hemoglobin, where one oxygen binding to one monomer, it's a tetramer system, hemoglobin. So one oxygen increases the binding affinity of the other monomer. So there is a cooperativity between these different domains. But again, that can be understood in terms of this hydrogen bond rearrangement also. Yeah.
Exact. And hydrogen bond rearrangement or many other things.
Yes.
What will be reinforcement is one thing which I think one of the, well, you have to think of it, that out of all these randomness, how much such order and stability, even dynamic stability arises.
Right.
So there must be some kind of attractor there in a physics, mathematics sense. Strange attractor kind of thing, that there are a lot of things are falling together to stabilize something.
Right.
And it is being done again and again. It is reproducible. It is not that it is happening once. It's same thing is happening 100,000 times, and they will probably do correct in 99,999 times. So there is tremendous driving force, and there's a kind of a funnel. And that funnel has been built over many years of evolution. And there must be a lot of gradient in free energy surface to hold it together. So there is very little stochasticity, room for stochasticity there. Okay.
But thank you so much, Professor Bakshi. That is so insightful. But allosteric states, they should be interchangeable with thermal energy, right? Because that is something that we, I guess, there's a broad agreement.
Suman's approach has been very good. You have to look at it from, look at problem from the end. It does not always help to look at from the beginning.
Yeah, since exactly, exactly that's what I was kind of mentioning in my end slide, that essentially you have very, very nearby substates which can interconvert with each other, right? So even like even in our traditional this kind of a protein folding model, there are local model globule states and things like that. Similarly, this is something we are working on. I didn't talk about it yet, but even in this when I talked about this kind of a contact map, there are interchange between, as I was saying, that there is interchange between one set of contact to another set of contact, but with rather low free energy barrier. So it's kind of going from one set of structures to another set of structures, and that can be tuned. It is based on the other perturbation factors. So actually in the free energy landscape, there are all these local minima, and it is interconverting between them depending on your external perturbation.
Yeah.
So that's the view that I'm suggesting.
Thank you, Suman. We'll chat again. Yeah.
Suman, if the barrier is so low, then they interconvert even normally, right, as a thermal effect?
Well, I mean, it is kind of pushing the, so let's say you have one set of population and you have one perturbation, then you're shifting the population. It's like if you have a kind of a toothpaste, let's say, I'm just giving a bad example, that it's a fluid thing, but if you push from one side, the fluid will go. And again, if you remove the pressure, it will come back, right? So it has to be mobile.
Yeah.
It has to be mobile.
Fluidity.
Yeah.
Yeah.
That fluidity is
Okay, go ahead.
important for adaptability.
Yeah.
So for adaptability, you should be able to, if you are too rigid, you cannot adapt. So you should be flexible so that you can kind of move based on the perturbation.
So each of them might be interconvertible thermally, but to have a direction like that, you need a perturbation from one
Right. So
end.
depending on the nature of the perturbation, it chooses one set of conformation.
Yeah, you break one hydrogen bond, you have to break many other hydrogen bonds because that particular chain becomes unstable.
Right.
So it is actually Feynman all the way, that Feynman plus evolution leads to the free energy surface, which free energy surface survived and which free energy surface is effective. There's a nice view of the connecting back micro to macro. There's a nice view.
So this, for example, this diagram I did not talk about, although I'll give you the idea that here actually we are using some kind of a clustering of all possible states. Now we can see actually very similar to a kind of a hierarchical free energy surface. So you can imagine that you see that all these lines here, they correspond to different states. Now there are groups. You see larger groups here. So this group corresponds to that normal wild type, and this is the phosphorylated state. But even in this bigger group, you see that there are substates, subgroups.
Your A marker is not working. I think you're trying to point it out.
Yeah.
But,
so yeah, anyway, so what I was saying is that here we are kind of clustering between all available conformational states, and there are two distinct groups. One is for the phosphorylated states, another is for the unphosphorylated states. But even within this bigger group, you'll see that there are subgroups, these clusters. So smaller clusters, smaller clusters. So this is kind of a hierarchical view. That very similar to like even your free energy landscape, there are a big basin. Inside a bigger basin, there are smaller sub-basin. And inside that, even there could be small ruggedness because of that individual torsional rotation. So we should have that kind of a hierarchical view of the conformational landscape. And
Sarika, now you try to close it. Huh?
Yeah. So I will, but there are questions, and let me.
I think, but we are already have 50. We have reduced to we have lost 70% of the population. And it's already, I think, 5 o'clock.
So maybe Suman, you can quickly answer the question. So if the hydrogen bond effect is made zero, we will expect no communication, is it?
In the examples that we have talked about, yeah, the hydrogen bonds are important.
Then next is, sir, why only hydrogen bond interactions are considered? Why not other type of non-covalent interactions are considered?
We have considered everything. So we are considering all the non-bonded interactions. And then we see that because of what kind of interactions, actually the energy is changing the most. And it happens to be, in this case, the salt bridges and the hydrogen bonds. They are contributing most to the energetic perturbation. But we are looking into all possible contributions. And
here there is a kind of a question. So phosphorylation is an example of intraprotein talk, example of long-range allostery. Like, is it an example of long-range allostery?
It is long-range.
Allostery means long-range in any way, right?
Yeah.
And Shushmita has a question. Shushmita, quickly, one question, ask. Shushmita, are you there?
Smita, I think she has left or something.
I will move on to the next one. Then, sir, is there any relation between proteins rather enzymes conformational changes? Shushmita, just wait a second. with its propulsion in its solvent medium, or during this conformational change does protein generate any mechanical force? Can you read the question, see what it says quickly?
We are not well, of course, any conformational force change will be associated with some kind of mechanical force, but we are technically looking at an equilibrium sample. So we are not doing any non-equilibrium change as such. So, but when you are doing this phosphorylation, yes, there are some stress generated. Whenever there is some perturbation, there is a stress generated, and that leads to the conformational change of the protein. Yeah.
Shushmita, quickly your question next.
Yeah, Shumanda, although we have heard this lecture earlier times.
Shushmita, make it short, make it short.
So, Shumanda, have you calculated the lifetime of these hydrogen bonds? Are they correlated?
Well, yeah, the individual, it depends on the system. For the PDZ domain, it's only side chain motion. So those are very fast of the order of the time scale of the network fluctuation is of the order of a few nanoseconds.
And have you found, next question, I just quickly, I got your answer. The second question I have quickly that are those hydrogen bonds that you found rewiring, are those conserved by any chance? Means conserved in a sense, I found that they are conserved in a sense that sometime like main chain to side chain, they are tending to be more conserved than the otherwise noises that
so let me just
Nilanjanadi was asking as well.
Yeah.
To
So when I said, okay, let me just say that when I say conserved, it is the residues, those are conserved, that which residues are participating, but it does not mean that that hydrogen bond will be there or not. So that state may depend on the perturbation, but same set of residues is that is conserved. That which set of residues is important. But then hydrogen bond can form or break.
Main chain to side chain.
Sorry.
Mostly are they main chain to side chain? Then the time scale would also be, means the lifetime would also be means making sense because.
Lifetime will depend on the strength of that particular hydrogen bond. There are some even side chain salt bridges. They have very long lifetimes.
Okay, Shushmita, I'll move on to the next person now. Now it is Shrabanidhi. How often do you encounter the presence of a salt bridge in case of allosteric inhibition?
Very often.
Okay, good short answer. Then it is Biman Jana who wants to ask a question.
Yeah, Sumanda.
Yeah.
For your first part, when you locked your loop and calculated the binding affinity, my question is that any particular loop conformational lifetime and signal traveling time. I mean, if you are locking, that has an infinite lifetime, but in
technically
normal...
what we did, well, actually we did not lock like that. We locked only by ligand binding when you docked later. But what we did, we kind of collected those conformations. So we have performed long simulations in different states of the loop, and starting with that, and the loop stayed there. And then for those conformations, we have computed the binding energy. So essentially, you are clustering the conformation, and we are grouping the binding energies together.
Okay, I got your point.
Okay, now the next question says from Premashish Manna. I find your mechanism of allosteric interaction without the structural changes, where you hypothesize the enthalpic cancellations at the local level can play a role, very interesting. How you can probe this experimentally? Do you find techniques like single molecule FRET can probe this?
In FRET, you need a probe probably, I'm not sure, but using solution NMR, it is possible to look at the distance distributions, and it is possible to check how the pairwise distance distributions are changing on the ligand binding. So maybe using solution NMR, it might be possible.
And now the last question is, can we detect the minute change by porcupine plot?
Yeah, so this is what I was saying, that normally, porcupine plot is in the context of principal component analysis, so we do that. But in my view, although in my view, a principal component is some kind of a dimensional reduction. It gives you a dimension, but it is difficult to physically interpret that what is that component. So that is one way of doing dimension reduction, but we prefer to look into in terms of this interaction network. That's just our point of view. We have done the principal component analysis, but I did not show those. Yeah.
Okay, good. Now there are comments which you will read anyway, because you know how to save it. Chats are there. And now it is over to you, Rajiv, to announce the next speaker.
Thank you, Sarika ji, and thank you, Sumanda, for